Hello, my name is Olivia Casimir. I am an upcoming sophomore at the University of Alabama in Huntsville, majoring in computer science. For my presentation, I will be discussing the automated process of quantifying scientific images using Fiji. For my introduction, for this experiment, I use Fiji, which is a software created by ImageJ to cut down the time it takes scientists to manually freehand and calculate the data in these rodent pancreas. And the rodents used in this following experiments are right here. And this is a logo of ImageJ, Fiji's logo. And for methods, step one, if you look at this picture right here, I use IJ1, which is a language created by ImageJ, so I can better understand the program to create more efficient results instead of using other things such as, or other programming languages such as Python, Java, etc. First thing I did was duplicate the image to find the color threshold. The color I used in this case is red. Then the program analyzes the particles in the new image. After the particles were analyzed, the program measured the particles for 1 pixel per nanometer. After it measures the particles per nanometer, then it collects all the data and numbers and lists out the measurements and the summary. Then it displays the results. And right here, as you can see in the duplicated image, that there are numbers isolates that were used to show which isolate is which. And it shows the summary right here and the measurements collected from the pictures. And for my key findings and results, I was able to automate the freehand drawing and calculation process of images using my program with IJ1. However, the only process that was not automated was the size and selection of the particles, which means that I have to individually go per picture to select a certain particle size to make sure the particles were selected as the best as can to cover the stained islet. Additionally, the program did not pick up the majority of the white space, which is actually beneficial to the experiment. An example of such is this chart right here shows a comparison between a fellow colleague of mine, Evann Fowler's manual version of the experiment and the computer-based version of the isolates being picked up, the area of the isolates. And the manual version is in blue, and the computer version is in red. As you can see, the computer version and the manual version have been pretty consistent, a slight variation where my area was a bit smaller. However, in this section as P8 and P10, which is known as picture 8 and picture 10, the difference was quite absurd. However, the reason for such was that my program was able to pick up the same colors for the islet and not the white space. However, if I can show you an example here, this was picture 17 used, picture 10 for rat 17, my bad. And as you can see for here, Evan manually drew out the section of the islet, while my program, however, only accounted for the stained parts of the original image. Another example is for this right here, this islet. Evan only counted the entire section of the islet that was stained. However, my program was able to pick up this more for the islet because of its particularly stained size, which showed that the program here was able to be consistent with Evan's work, but in this case right here, it was not consistent due to the white space. For impact and conclusions. In conclusion, I was able to automate the process to help scientists cut down the time for manual work to help them find the solutions needed for research and development. However, even though I was not able to automate the individualized process of the pixel sizing, I was able to cut down a significant amount of time in finding particular stainings of the islets and the calculation process. For my acknowledgement, I would like to thank Evann Fowler for allowing me to use his data for this project and Dr. Vogler, who allowed me in this research, and Ms. Wimalarathne and Mr. Cook for their assistance, as well as Alabama Lewis Stokes Alliance Minority Participation, NASA, and Alabama Space Grant for their contribution. Thank you.